Good morning, everyone. I am Dr. Rituparna Saha, Assistant Professor in the Department of Life Sciences, School of Biological and Forensic Sciences at Kristu Jayanti deemed to be University, Bangalore, Karnataka. Today, I will be discussing a fascinating topic that bridges physics, chemistry, and biology: radioactivity. In this session, we will understand what radioactivity is, how it occurs, how it's measured, and some of its important applications in science and medicine. What is radioactivity? Radioactivity refers to the process by which the nucleus of an unstable atom loses energy by emitting ionizing radiation. This phenomenon occurs because some atomic nuclei are unstable. They have an imbalance between protons and neutrons. To reach stability, they release energy in the form of alpha particles, beta particles, and gamma rays. This process, called the radioactive decay, continues until a stable nucleus is formed. Radioactivity is a natural phenomenon. It occurs in elements like uranium, thorium, and radon, but it can also be artificially induced in laboratories. Let's now look at radioactive decay more closely. Radioactive decay is a spontaneous process. It doesn't depend on temperature, pressure, or chemical reactions. There are mainly three types of decay: alpha decay, in which a nucleus emits two protons and two neutrons, reducing its mass number by 4; beta decay, in which a neutron gets converted into a proton with the emission of an electron; and gamma decay, in which the nucleus releases energy as gamma rays without changing its atomic number. Each decay type results in a different daughter nucleus, gradually leading to a stable form. These are some of the examples of the three types of decay that takes place. To quantify radioactivity, we use specific units that measure the rate of radioactive disintegration. The Curie is one of the older units, named after Marie and Pierre Curie. One Curie represents 3.7 into 10 to the power 10 disintegrations per second. The becquerel, which is the SI unit, represents 1 disintegration per second. And another unit is the Rutherford, which equals 10 to the power 6 disintegrations per second. These units help standardize how we measure the activity of radioactive substances both in research and industry. One of the most important concepts in radioactivity is the half-life. The half-life of a radioactive substance is the time required for half of its atoms to decay. It is a constant value for a given isotope and is not affected by external conditions. For example, carbon 14, which has a half-life of about 5,730 years, makes it useful in dating archaeological and geological samples. Understanding half-life helps scientists estimate the age of materials and determine how long radioactive isotopes remain active. When it comes to detection and measuring radioactivity, there are a few key devices that we use. One of the first devices to be used was ionization chamber. It is one of the most simplest and most reliable instrument that is used to detect and measure radiation. It operates on a straightforward principle that when radiation passes through a gas-filled chamber, it ionizes the gas molecules, producing positive ions and electrons. These charged particles are then collected on oppositely charged electrodes, creating a measurable electric current. The magnitude of this current is directly proportional to the intensity of the radiation. Ionization chambers are mainly used for measuring high levels of radiations, especially in radiation protection surveys where accurate quantification of exposure is very important. Another device which is used to detect and measure radioactivity is the Geiger-Müller counter, which is sometimes simply called a Geiger counter and is probably the most widely recognized radiation detector. It consists of a gas-filled tube with a central wire electrode and operates at a high voltage. When a radioactive particle enters the tube, it ionizes the gas inside, creating a brief pulse of current. This pulse is then amplified and is registered as a visual signal on a meter. Each visual signal represents a single ionizing event, meaning it counts individual radioactive emissions. Geiger counters are extremely sensitive to beta and gamma radiation, and to a lesser extent, alpha radiation. Scintillation counter. The scintillation counter is a highly sensitive instrument that is used to detect and measure very low levels of radiation. It is based on the principle of scintillation, where certain materials called scintillators emit tiny flashes of light when struck by ionizing radiation. These light flashes are then detected by a photomultiplier tube, which converts them into electrical pulses that can be measured and counted. Scintillation counters are capable of distinguishing between different types and energies of radiation, making them more versatile than Geiger counters. They are widely used in medical imaging, biochemical assays, and radioisotope research, especially when precise measurements of radiation intensity or energy are required. In fields like nuclear medicine and environmental monitoring, they serve as crucial tools for detecting minute quantities of radioactive substances. Radioactivity also has several beneficial applications, especially in medicine and biology. In medical diagnosis, radioisotopes such as iodine 131 are used in imaging to study organ function, for example, thyroid scans and bone scans. Positron emission tomography uses radioactive tracers to visualize organ function in the body. In case of radiotherapy, isotopes like cobalt 60 and cesium 137 are used to target and destroy cancerous cells through controlled radiation exposure. When it comes to sterilization, gamma radiation sterilizes medical instruments like syringes and surgical tools without using heat, which helps maintain their integrity. And in case of tracer studies in biology, isotopes like carbon 14, tritium, and phosphorus 32 are used as tracers to study DNA replication, enzyme mechanisms, and metabolic pathways. Radioactivity also finds application beyond medicine. In cases like radioimmunoassay, it's a highly sensitive technique that uses radioactive isotopes to detect and quantify hormones, drugs, and proteins in biological samples, even at extremely low concentrations. For environmental and ecological studies, radioisotopes helps to trace nutrient cycles, monitor pollution, and study energy flow in ecosystems. For example, isotopes can reveal how pollutants move through soil or how plants absorb nutrients. These applications highlight how radioactivity, despite its potential hazards, play a crucial role in advancing science and improving life. To summarize, radioactivity is a natural process that helps us understand atomic stability and energy transformation. We have seen how unstable nuclei emit radiation, how their decay can be measured, and how these phenomena have been turned into valuable tools in medicine, research, and environmental science. While radioactivity must always be handled with care due to its potential health risks, its applications have transformed modern science and healthcare. I hope this session has helped you gain a clear understanding of the concept and applications of radioactivity. Thank you for watching.